Hello, welcome everybody. Thank you for joining us one more week. We are super glad to have you here today. And very briefly, just an introduction of our platform. If you haven't been here before, perhaps you have heard this introduction many times. PhenomeForce is a platform that was created a couple of years ago to share resources in plant phenotyping. We have been covering many different aspects. This year, we are covering root phenotyping, which, as we know, is an emerging and quite interesting field for plant phenotyping. And if you want to learn more about us, or do you want to see previous recordings, you can learn much more details about our platform in just a second. You can, sorry about that. You can find all the details in our website, or just follow our Twitter, because we keep posting quite often to announce the new workshops that are coming, but also to share interesting links. All the recordings from all the previous workshops and the one that we are about to get started are available on our YouTube channel for free. And if you have any questions or any suggestions or any kind of comment or want to get in contact with us, please use our Gmail account or join our space in Slack. And with that said, Maria is gonna introduce our next speaker. Oops.
Hi, everyone. I just would like to welcome our today's speaker, and Dr. Guillaume Lobet. He's an assistant professor at Forschungszentrum Jülich and UC Leuven in German and Belgium. And today he will be talking about using functional structural plant models to extend experimental phenotypic knowledge. And Dr. Lobet, thanks a lot for accepting our invitation today, and all the material that you will need for follow-up with this presentation will be in the chat. And with that said, I would like to hand it over to Dr. Lobet, and we will be here on the background assisting you with everything. Thank you.
Thank you. Thanks for the invitation. So hello, everyone. So it's a pleasure to be here, and I hope you will enjoy this session. So I just have to figure out how to share my screen, and maybe asking for help from the background. No.
Hey.
Hey.
Hi everyone. Guillermo, you already sharing your screen. Just put the main presentation over there. See, it's already there.
Okay. So now it's there. Okay.
Yeah, it is there.
Perfect.
Just put your slide. Here we go.
Okay, perfect.
Good luck.
So is that fine? Thank
Yes.
you. Okay, so as was said, today I will try to show you how we can use computational plant models, so what we call functional structural plant models. I will explain that a bit more today, to use that in combination with phenotypic data. And what I will try to do is actually to show you how that can be useful. You can, I put a link there. That is also, I think, in the comments. On that link, you will find all the links in the material that we will use today. So you can go there. It's also in the comments if you want. So what we will do in this workshop, I will try to show you, will make you discover how this type of models, so these computational plant models, can help you extend your phenotypic data in two different ways. So from phenotypic data to more phenotypic data, but also from what we would call structural data to functional data. So try to make the link between these two, and in both cases, the model will be the kind of tools that we use to make this link. And then I will also, if we have time, discuss how we can link then experiments and models, and what you should be paying attention to when you want to do that. So if after the talk you say, okay, I want to actually use the models, then reflect on how you should think your experiments to be able to do that in the most efficient way. So one thing or two things that we actually not do is this session will not be an in-depth hands-on with the models themselves. So we will use the models, we will use actually web interfaces of the models, which are kind of easy to use, push button version of the models that we actually never use for research purposes, but in the case of the workshop, will give you a good feeling of what the model can do. So I will not explain to you how to install the model on your computer or all the really needy of the modeling itself. What we do is show you why it can be useful. And if you think it is, then you can think more about installing the proper version of the model. But we will not need that for this session. As I said, we will use online models that are very easy to use. Also, it will not be an in-depth hands-on with image analysis. So at the end, I will show some image analysis, but I will really focus on the link between the image analysis and the model. I will not spend a lot of time on the image analysis itself. Okay, so it will really be more a conceptual talk on what we can do, how it can be useful to do this link between the data and the model. And for that, we will use tools, easy ones, but I would say not the research grade versions of the tools. More, that said, the results that we will get are the same that we get with the models that we use. It's just that the interface are a bit different. Okay, also all of this will also, I maybe not mention that clearly, but we'll focus on roots. So we will work on the roots. So it's really in the continuity of what the previous workshop from Benjamin and Abraham. We will stick with the roots and look specifically on that part of the plant. And so the first part of of this workshop, I want to focus on how to get structures, how to move from some structural traits to better structural traits, or to more structural traits. And so really a focus here on the root architectures. I will also do give some examples with root anatomies, but we will not. There will not be exercises on that one. We will really focus more on the architecture itself. Okay, so the first thing, and again for the ones of you who have been to the previous workshop, or for anyone who has worked with roots altogether, we know that root phenotyping is complicated. It's complicated because you always have to choose a method to observe your roots, which normally are growing into the soil. And every single method comes with compromises that you have to make. Compromises usually between what I call here on this graphic, the naturalness of a setup and the throughput being being either, is it a high throughput? Is it high throughput setup? Is it a deep phenotyping setup? So always the trade-off between what you use, how natural that setup is, and then with that one, how much information you can get and how much plant you can observe. What we see here on this graph, as I said, on the x-axis is what I call here, which is a made-up word, which is the naturalness of the setup, which is basically, again, a kind of indices that is a mix between whether the roots are growing in 3D or 2D, whether there are local constraints to the growth of the root. So for instance, in aeroponic setup, when the roots are growing into mist, there is no local constraints to growth, and it's a 3D setup. Whether you have an infinite growth, so whether the root is contained or not within a container, and whether you have oxygen or not, and whether it's in soil or not in a soil. So what you see on the very far right of this figure are actually field setup. And then on the right is field setup, and then on the left is very artificial setup, such as petri dishes, pouches, aeroponic, and so on. And usually what we see is that if you want to have a high throughput or deep phenotyping setup, usually you have to move away from the naturalness of your setup. So that's really the compromise that I'm talking about. That's one thing. So it's complicated to observe the roots, but then even when you observe them, there are actually many ways you can quantify a root system. So here I'm not talking really just about individual roots sitting into the soil. I really want to talk about root systems. So this assembly of many different roots composed on different types of roots. How do you quantify that? And that again can be done in a lot of different ways. And the few slides that we show here are actually coming from a teaching tool in plant biology that we wrote together with Larry York a few years ago, that you can also find online. And so here I just want to show one type of classification that you could make between the type of information that you can get out of root system. So the first one is what we call the geometrical information about your root system. What do your root system look like in space? And for that type of information, you basically need the position of every single segment of your root system. And that will give you information such as the depth, the width of your root system, potentially the convex hull, if you're interested in that type of traits, length profile, you could divide that. And so these are very synthetic metrics that you can get out of it. The thing is, these could be, in some instances, misleading. So if I take the example that I have here, so these two root systems that I'm showing here, they actually have an identical width and depth. So in both cases, if you're looking only at this type of global metric, they could look the same because these metrics are the same, but they are actually very different root systems. So these metrics usually are not they're not enough if you want to know what your root looks like. Then you can move one step forward, and it's looking at what we call the morphological information. What do individual root looks like? And that can can take the form of diameters. So different roots have different diameters, for instance. They have different length. That requires usually a bit more time to analyze your image, because then you would need not only to know where the roots are, but then you would need to actually take measurements on these roots into your image. That will mean tracing the different roots and getting the diameters, getting the orientation, and so on. But that information is already carrying a bit more, is a bit more useful than just these global type of metrics. And here, what I want to show here is that if you could actually have a root system with very different morphological information, which is what I wanted to show here. In this case, what I have is two root system with a different growth rate of the primary root. So the red root has a different growth rate, but also the distance between two lateral roots is smaller. So here we have two root systems that look very different because we have two key development process growth rates and the branching that are different, but they actually have the total same length. So in that case, again, looking at just one metric, the total length of your root system would not be enough here to distinguish these two biological objects. And that's where you can, again, move a bit forward. I'm actually mixing a bit here the different type of information, but one step further than just the morphological information, which is again the length of the different roots, is including on top of that, including the topological information. How are the roots connected between them? So do we have a primary root and then natural roots? Okay, how do we have, because again, once you have that, then you can make classification between your roots. Different roots might have different properties, might have different functions. And if you have just the length of every single root, but you don't know how these roots are related to each other, then you cannot do the difference between that. We'll also show later on why that might be important. One root is not another one. Okay, so we have different types of roots that have different functions within your root system. So topological information can be the number of laterals that you have, the distance between these two laterals. Another one that that is often used is what is called the LAUZ that you see there, which is the length of the unbranched apical zone, which is the distance between the root tip and the first lateral, for instance. Okay, so but for all of these type of more fine information about your root system, then you need to know the connections and the explicit relations between the different root orders. So again, every step, it's a bit more difficult to actually get this information because you need to get more and more data about them. And then the last one I want to show is dynamic information. In that case, it's not just looking at one root system, but looking at the root system across different time. And then that allows you to have dynamic traits, such as emergence rates. How fast are the laterals coming out of this primary root, and how fast they are coming out, but then how fast they are growing. How fast is the primary root growing? How fast it's growing not only along the axis, but also the secondary growth. Okay, so there is plenty of different dynamic information that you can get. Again, this is getting more difficult to get experimentally because you need to follow the same root system one time step after the other. But what I want to argue is that as soon as you have dynamic information, if you have also the topological information and the morphological information, then you have all the information to actually look at your root system. The thing is, it's sometimes hard to get all of that. You might also not know which one you want to get. Okay, maybe it's not important for you to have the topology. Maybe it's not important for you to have the dynamic information, or maybe you can find your way around. And that's always depending on the question, the scientific question that you are actually that you want to address. And what I want to show now is actually how you can sometimes use models to make links between these different types of information and maybe rank or look at the information that you might be more interested in. And so what I want to show is how to use, again, these computational plant models to extend the data set that you might have acquired experimentally. So what you can see here is a very simple view of what can be done with that. Is if you have a computational plant model that will take a certain amount of parameters, okay? Usually parameters that are process-based data. What I mean by process-based is dynamic data, such as growth rates. The models that we use are actually dynamic models. They are models in which you will have a root that is growing from a seed. And so you need dynamic parameters that are linked to process, such as branching, growth rate, and so on. And then from that, if you enter this data into the model, then you can get more global data. The model can tell you, okay, if I have an idea of the growth rates of my roots, you input that into the model will give you an idea of the total root length of the exploration of the soil by your root system. And what we see also after is a link with functions. We will see that different types of root systems that might look alike might actually have very different functions. And we will try to make the link between all of that. And for that, the first tool that I would like to use is actually a model that we developed in our group at the Forschungszentrum Jülich. Forschungszentrum just means it's the German word for a research center. So it's not very complicated. But it's complicated to pronounce. It took me a month to be able to do it more or less correctly. I'm not even sure I do it still, but it just means research center. That's what I meant. So we develop a model, computational plant model that was first called cRootbox, and what we use now because it's just simulating the root system. Now it's actually a full plant model, so we could see plant box. So it simulates the full plant. That model is actually open source, so you can find it online if you go on that address. No, if you type cRootbox in your research engine, you will find it. That address here actually leads you to a web interface of the model. So the model cRootbox, it's a model that is coded in C++, which makes it very fast. And around that C++ code, we actually have a Python layer, and that Python layer allows you to interact pretty easily with the different functions of the model. You don't need to know C++ to use the model. Usually it's in Python. But then what we also did was set up this web interface that I will show you right now. So the web interface looks like this. What you have to do is just at the beginning, just select the one plant that you want to simulate. So we have a set of predefined parameter sets for cRootbox. So we'll take the first one, Anagalis femina. And then you just click here the button, Unleash cRootbox. So what happens when you do that is actually you just launching the model in the background, okay? So if I just click again, you will see that it's a different root system every time, because every time it's not a precompiled root system, it's a root system that is generated on the fly. So again, the C++ code is very fast. So when you click the button, you run the model on the server, and then you just display it like here. And then what you can see is a synthetic matrix, such as, for instance, here, the total root length of your root system, the number of root segments that you have, or the maximal depth. Basically, once you have your root system structure, you can get whatever information you might be interested in. And then what the web interface allows you to do is actually to change the parameters of the model, and change the parameters and see the direct effects of changing these parameters. For cRootbox, again, I will not go too much into the details here. That's not the point. But cRootbox actually takes parameters for every type of roots that you want to simulate. So again, the root system is made of different types of roots. So what you can see here on this representation is a red root, which is your primary root, your first order or zero order root. And then you have lateral roots here in green that shoots out on the side. So these green roots. And you can directly see that they already have different type of parameters. So these green roots, you can see that they have a tropism that is not, they have a plagiotropism. So they tend to keep horizontal when they grow, while this red one, it tends to go downward. So it has more gravitropism behavior. And so each of these colored roots, so these four types of roots here, they will have different types of parameters that you can change with the model. And that's what you can see here. So depending on the type of plant that you select here, so we'll just select, let's say another one, Brassica napus here. We'll run it again. Well, that one is not really nice. Let's take another one. That looks nice. Okay, this one looks nicer. You can see here that's actually the name of the root changes. Okay, just because the name of the roots in the parameter file are different. Okay, I will go back to Anagalis. That was just to show you that you can simulate different types of roots. And then depending on the type of root system that you're simulating, you will have different type of, the number of types of roots that you have will change. And then for each type of root, you have a set of parameters that you can see here. Okay, the main one, the one I want to show you a bit are, let's say, the growth rate of the root. So root will have a specific growth rate, and will have a maximal length. So usually roots tends to have a maximal length. So not all the roots will grow indefinitely. Some of them will stay within a maximal certain range of length. And then another parameter that is very important is also this interlateral distance. So that's the distance between two laterals on a single root here. Okay. What we'll do now, and so, no, sorry, just show you one more thing. So if you select one of these, okay, again, I'm not going into the detail of all of them, but for instance, here we just change the initial growth rate. So that's the growth rate of, in this case, the primary root. If, let's say, I would just divide it by 2, and then I update my root system. So I'm not clicking here on unleash, but on updates. That means that I will regenerate the root system, but with the parameter that I changed. And you can see here that the total, the length of my root system was divided by 2 in this case. Okay, so here is already one type of effects that you can, where having a local information about your root system. In this case, I have a local information about the growth rate of my primary root. Okay, I know that, let's say, between two genotypes, I know that one of the genotype has a growth rate that is the half of the other one. Then the model here can help me understand what that means in terms of development on a longer term. If I have a growth rate that is half as long, and if that growth rate is the growth rate of the primary root, that means that altogether I will have first the primary root that is, again, half as long, but I will also have less lateral roots. And how does that translate in terms of total root length? As you can see here again, so here we have total root length of 300 centimeters. But that's not the length of my primary. My primary is only 20 centimeters here. But if, again, I will double it again, so go back more or less to the normal, my primary is longer this time, but I can see that my total root length has increased much more. What I want to show here is that because the different processes, developmental processes within your root systems, they are linked together, you don't produce a lot of laterals if you have a very short primary. So because these are links, taking measurements, some measurements in your root systems that you can then feed afterwards into the model. So again, in this case, I'm just measuring the length of my primary. The model can help me understand what will be the effect of these local small changes on the global architecture of my root system. And then for that, just to again show you kind of illustrating that with a few examples, what you can do right now, you can do in the same at the same time as I do it. Again, you can log in into the web interface. Actually, not sure how many of you are online right now. It should not crash. I think that if there are 200 persons logging in on the web interface, it might crash. It should not be the case. If it does, then I will ask you, some of you, to unlog just for a while so I can do the exercise. But then the exercise basically is just to observe what is the effect of a change in the growth rate if you change for the primary. So I just showed you right now what is that effect of the primary. It does not only affect the primary root, it also affects the other roots in your root system. If you do the same changes, so let's say divide by 2 the growth rate of your first or the lateral, is that the same? Does it lead to the same change, for instance, in the total root length of your root system? And is it also the same for the different species? Okay, you've seen that we can actually change the species here. If you change the species, is a change in parameter. Again, parameters that you have measured experimentally. Does that have the same effects on the development of your root system or not? One last thing, you can actually start playing around with the model if you want. I will continue explaining things, but feel free to test it. One thing here is that what we see here illustrated on this web interface is actually the final point of the simulation. So we don't see here the dynamic aspect of the simulation. Okay, but in reality, the simulation is not just simulating one root system like that. It's actually starting with a small root here, a small red root that will grow and that will produce a root system that is larger and larger. To illustrate that, what I can show is change one of the parameters here, which is the length of the simulation. If I actually choose a smaller simulation, you can see here that at the beginning, we wouldn't go smaller than that. At the beginning, I just have one root with one primary, very few laterals. And then as you go on, then your root system is growing and getting more and more complex. Okay, but what you see there is only the end time point of that root system. Okay, so just back to the question that I asked, which is what happened if you actually look at the same parameter, the same change, in this case growth rate, but that change in growth rate happens on different root types or in different species. Is that effect always the same? As you might expect, the answer is no. So again, I will show here. So here we have a growth rate of 671, total root length, sorry, 671 centimeter. Just for the sake of remembering it, I will write it down here. So 671. Okay, so let's say that's my base simulation. Now, if I just divide the growth rate of my primary, now I go down to a growth rate of 410. But that's not the growth rate, sorry, total root length. Actually, if I run the root system a couple of times, you will see that we have quite some variation in that number, okay? That's because for each of the parameters that you can actually have a standard deviation for these parameters. So some parameters do have a standard deviation. Here it's 0, but it's actually not the case for all parameters, which leads to some stochasticity into the model. What we're just looking at right now is just, I would say, big differences. Basically, if you decrease the growth rate here, it has, of the primary, it has quite an influence on the total length of your root system, again, because shorter primary means also shorter laterals and so on. If now I do the same, but let's say for the first order laterals, I decrease that. Again, you can see here that we have 10% deviation, which means that we have quite some variation. I will actually put that to 0. Here again, the effect is different. So I have, I will just put back the primary root to 4. Okay. Oh, it's actually not so much difference in this specific example. 301. And if I change that one, yeah, okay. Now, if I change again, I will look at another one. I'm looking at the second order laterals here. We'll again decrease the growth rates. Here we don't see much changes. That's because the maximal length is actually already quite short, so they cannot grow much. Now, if you look at another parameter, let's say if I want to look at not here the growth rates, but the interlateral distance. Again, the interlateral distance is the distance between two lateral roots. That one will also have a very strong effect on the total length of your root system. So for instance, here, what I will do is on the primary, change the interlateral distance to 0.4. I update my root system. I have much more roots. And here you can see that it's more than double the amount of the total root length that I have. So it was 140 something. 1471. So what you can see here is the same range of changes divided by 2 on either different types of roots or different parameters, and will also be the same for different species. So if I look here, I would take this cabbage here. If I look at the main root parameters here, because I have several of these, no, I will not take the main, several of these main roots. If I change the growth rate divided by 2, so the total root length here is quite long, so 24,000. Oh, that's actually perfectly divided by 2, which is random chance in that case. Well, actually not so random. So on the primary, it's not only by chance, because you just reduce the size of all the root system. It should not be the same if you look at another type of roots. And there we don't see much difference again, probably because the maximum length is different. Okay, so what I want to say here is that, again, here in this case, because there are too many links between the different parameters. But basically, what I want to say here is that depending on the parameters that you change, and depending on what you measure in your experiments, the global change of what you measure, the global change induced by this change in parameters can be different depending on the roots that you're looking at, the parameters that you're looking at, or the species that you are looking at. Even though here, in the examples, some of the examples that I show, that was not completely obvious. But what I wanted to show here also is that if you don't need, for instance, to have measured the total root length of your root system. You could actually measure some of these dynamic parameters, such as the growth rates of the different roots, or you can measure the topology of your root system, and then you could actually use the model to extract the total length of your root system. In some setup, getting the total length of your root system is not possible. But it might be possible, and we'll come back to that a bit later, it might be possible to get some parameters. If you have only a subset of the parameters, the model can help you then from this subset from these parameters, actually get more of these global data. Okay, is there so far? I don't hear anything. The questions? No. Okay, moving on then. Moving actually to the interesting part, which is, getting information about functions from structures. What I mean by function is, when you're doing an experiment and you are actually measuring root traits, ultimately what you are interested in is the functions of these roots. If you are measuring the total length of a root system or the depth of a root system, is because usually you are interested in the uptake capacity of that root system. If you are measuring these structural traits, they are usually because you think they are a proxy of a functional trait. What I want to show here is how these link between function and structure can be done again with the models. And I will take the example of water, because that's a topic that we work on quite a lot in the lab. So for water, just a brief biological reminder, water flow in the soil plant atmosphere system, it's a passive process. Okay, so there is no transporters of water within the plants. The water moves from wet compartments to a dry compartment. The drier compartment in the system being the atmosphere. And so as soon as the plant opens the stomata, what happens is that water gets out of the leaf into the atmosphere, because the atmosphere is much, much drier than the inside of the leaf. And then that creates a tension, and that tension goes from the leaf to the stem to the roots, and then from the roots to the soil. And that triggers then the uptake of water from the soil into the root system. Very passive process, but that is regulated actively by the plants. It's regulated at different levels. First, at the levels where the water is entering into the roots. So how easily can the water enter into your roots? And that depends on a lot of different factors, anatomical factors that we will come back to later. Anatomical factors that can be the number of cell layers that you have, the type of cells that you have, whether you have hydrophobic barriers, so Casparian bands or suberin deposition within your root system. Depends also whether you have aquaporins, that are these water channels that can be actively deposited by the plant, and so on. So a lot of different factors affecting that. Also factors affecting the axial conductivity, which is how well the water can go up inside the roots. Again, usually it's the number and size of the xylem vessels that you have. And then an important part is also the architecture. So the architecture of the plants, the root system, defines in large part, not everything, but in large part, the access of the plants to water. Okay, if you have only one small root, you have less access than if you have a very long root system into on your plants. So the architecture is one very key parameter here, and that's the one that we will work on right now. You also have the soil that will influence the uptake, mainly because as the soil dries, it becomes less and less conductive. So if the plant is taking up all the water around it, then there is no water available anymore for later stages. And so that creates kind of its own, and create its own stressful environment. We will not talk about that. We'll mainly talk here about architecture. What we use in our models to represent the water flow within the plant, the root system, is what we call an electrical analogy. So it's a very simple physical relationship that defines a flow between two compartments. And that flow is defined by the difference in potential and by the conductivity between these two compartments. So as I said, you have different in potential between the soil and the roots, between the root and the shoot, between the shoot and the atmosphere. And between all of these compartments, you have several resistances that can change depending on the conditions, the age of the plant, the type of fruit, and so on. So we have, that's the global, let's say, relation that we use to represent water flow. And then what we do is we actually apply that equation, so that flow equation, we apply it on every single point in our root system. So every single root segment is connected to the soil and is connected to its neighbor. And we can then compute the flow of water between that segment and the root, and that segment and the segment downstream and the segment upstream. So if we use here the computational model, we can reapply that sort of equation in every point in a root system, and that will allow us to actually resolve the water flow within the whole structure. Again, I will not go into the details here of that specific resolution. From that, we can actually compute a metric, a global metric that we call the root system conductivity. The root system conductivity is the conductivity of the whole root system. It's not the conductivity of a root segment, a single segment, but of the whole thing. And that defines, to some extent, the intrinsic capacity of the intrinsic uptake capacity of a root system. Okay, if it's large, that means that the root system has the capacity to uptake a lot of water. Again, if I'm taking the example of one single root, that one single root will have a very low root system conductivity because there's only one root. If you have thousands of roots, then that root system conductivity will increase quite a lot. Here is an example of the type of results that you can simulate with this type of models. You can see on the left, what you can see is actually the growth of a root system with the model C root box. So that shows you that it's actually really a dynamic model. And then if you use, again, these equations, these flow equations, and then you resolve that on a root system, you can put your virtual plant into virtual soil and simulate the water flow, the uptake for all of these different plants. And you can also simulate specific behavior as the one you see here, where what happens here is that actually the water, the plant was taking up too much water for that specific soil, up to a point where there was no more water left in the soil and the plant was forced to stop taking up water, close the strata, and stop the transpiration. So this is the type of results that we can get. What we can also do with that is, because it's a model, we can actually generate a lot of different combination of traits. Okay, so we can vary all the different traits of our models, which generate a lot of different, let's say, phenotypes of your plant. Then we can test these different phenotypes. What you see here is precisely that. So every point is one phenotype. In this case, what we did was specifically try to explore the phenotypic space as possible. That's why you have this dense cloud of points. And for each point, what you see is the relation between the root volume. So it's really the volume of every single root, and the relation with the cumulative water uptake. So basically what you see is, do I have a big root system or a small root system, which is that is the root volume, and whether that root system is able or not to take up a lot of water. And so we defined in that figure the efficient root systems as these red ones that you see here. And these red root systems, they actually have a low root volume with a high water uptake capacity. Because these are what we defined as the efficient one. Again, here, by combining all of these traits with the models, we can actually test which combinations will be the most efficient. We can also see that it's not necessarily the same in the different environments. That's actually what we see here. So here in these radar plots, every line is one simulation. And you can see the gray line in the background. That's all the simulation from the dataset. The blue lines are the simulations, the efficient root systems. And what you can see is that we don't have one specific combination of traits that is the most efficient. You have many combinations that can lead to the same efficiency, which is a way to say that there are compensatory effects between the different traits that you might measure. Measuring one trait is usually not enough to actually get a good idea of how efficient a root system can be, because maybe one specific trait might not be, might actually lead to a lot of different efficiencies. Okay, just before I move to the next exercise, where we will play a bit with this concept, can I just get an audio feedback just to be sure that there are still people on the other side from the organizers?
Hello.
Yes, perfect, good.
Hello, yes, we still have quite an audience. I was waiting for you to make a pause.
Okay.
Because we have a question on the possibility of using all these tools with wheat roots, which is also my interest. So I assume it is possible to use it with roots, but
yes.
And I have a second question for you afterwards.
Yeah, okay. So short answer, yes. I think we might actually, do you still see my screen? No, you don't.
No.
You don't see my screen, okay. I'm just getting used to the system.
But we can.
So read root, no, it's fine. You don't have to wait. So cRootbox is meant to be a generic model, so we can simulate a lot of different types of roots. Wheat being one of them. We actually have right now in our group several projects on wheat specifically, so we can simulate a wheat root system. Mm-hmm. Normally, you can simulate any type of root system, but that might require different types of parameterization.
Excellent. And my question is fantastic that we can actually predict the water uptake. And this has quite important implications for many projects, particularly in my case, I'm really into looking at the impact of the root structure of wheat into soil structure and things like water holding capacity and aggregation. So the idea of also linking it to the water uptake, to which extent do you think it will be actually possible to link it to the actual soil structure in things like the water retention capacity, because obviously it's linked to the uptake of water.
Yeah. Okay, so in general, so what I show here could be applied to any type of questions, with the restriction that there need to be a model that take that into account. So I'm not aware of a model that actually have that feedback between root growth and soil water uptake capacity. That does not mean that it does not exist. I'm just not aware of it. Okay, so that's that would be my bottom line. If the model exists, then you can use it in that way, I would say.
Or we will need first to collect the data and build a model?
Oh yeah, that's the long way. Yes. That's
so I suppose at the end of at the end of the day, the question is because there was a question in the previous comments about how to build the models with data. So the start is just once we have a good set of data, we can generate pretty much a model to predict anything these days.
Yeah,
just need to start by collecting but the limitation is the data. The models are not a limitation, so we are in a position to build models and connect any plant phenotyping with actual parameters in soil and the activity.
Yeah, so that's so you can definitely always build a model if you want. Usually it's a lot of work. So for me, the first question would be to really look around whether there are models existing, and I will actually put at some point a link to a website that reference a lot of plant models that exist out there. And then even though if something is not completely what you want to do, look for the closest thing. So look for the model that you think might be the closest one to the topic that you are interested in, and then talk to the people developing that model.
Yeah.
Usually that might be much faster than doing the model from scratch.
And also not everybody has the background to build a model. So the bottom line is that there are plenty of models available.
Yeah, and then it's not always necessary to build the model. I mean, the models can answer some questions, but not all of them. Sometimes it's not needed. And there also, so when I say model, there are many types of models. Again, it's not the place here to discuss all of that, but there are many types of models that you might use. And each type has its own strengths and limitation and type of questions that you might be able to answer.
Well, it's up to you. We have still a few more questions in the file, or would you like to continue and say?
No, I see. So here the question is low volume, high water use should also include surface area. Yes, indeed. Oh.
Sorry.
Oh, that's neat. Am I the one doing that? No, you're doing it. Okay. I'm discovering the interface. It's very nice. Yes, of course. But when I talk about volume, it's actually closely related to surface area. So it's the sum of yeah, we could actually do surface area instead of volume, but I would expect it's very closely linked. So just to be sure, I'm not talking about the volume occupied by the root systems. It's not the convex hull volume. It's really the volume of the roots. But that's a very accurate comment, because what is important is the amount of roots, the surface of the roots touching the actual soil.
Right.
Okay.
There was also a question on from the registers, from the attendees. How can root phenotyping be applied to understand mechanical properties of roots involved in soil reinforcement and erosion control, which is a bit linked with what I was saying before?
Yeah, again, I think for that specifically, the models can help you. So physical model of soil physical properties. There are models doing that, in which then you can insert a root-like structure with certain physical properties. Again, it boils down to, do you have a model that can help that represent that specific process that you are interested in? If you have, then you can use the model to at least try to get some information about your question. In our case, we are interested in water uptake, so we have implemented a mechanistic representation of water uptake within the model. We have implemented physical laws of water flow between different compartments, and because we have that, then we can just run the model by varying all the different parameters and try to understand exactly how the system works.
And that will apply also for another question that was on metabolomics. Like,
Yep.
you know, some people, the approach to root phenotyping is metabolomics. If it will also be possible to link it, it's along the same lines, I suppose.
Yeah, it's on the same line. I would say, so our specific models that I'm showing right now, they don't include metabolomics. So these are not the right model to do that. But in general, if you have a model that can do it, and for metabolomics, well, it depends on the scales that you are looking at, of course. But yeah, again, you just need to have a model describing that phenomenon. And ideally a mechanistic model. So a model that will represent the processes leading to the development, not so much a statistical model that is based on pre-existing relations between the different parameters.
Yeah, that makes sense. And if you don't mind, just a last question,
Sure.
and then we should continue. It's something that keeps coming in every workshop. There seems to be a lot of interest in root phenotyping in trees. And of course, there are things like how to approach that, with the time scale of roots in trees, and also the fact that you have to sacrifice a full tree for some of the approaches for root phenotyping. Do you have any advice on this?
Yeah, so there is some But quite some papers on tree phenotyping, tree root phenotyping, and also on tree root modeling. I would say that one of the use of the model again could be to extend a limited data set into a broader one. So, for instance, one of the difficulties with trees is this multi-year cycle that you have, and so your tree is always growing and growing and growing and growing. One thing that you could do with the model would be to actually parameterize your model at the beginning of the life cycle of the tree. So you're not using a full tree, you're not measuring young ones, and then you let your model runs and see how that develops. So you use data from the beginning of the life of the plant, and then you use that data, you use it in the model to go further on. The issue is if you do that, the uncertainty of your data, of course, can be quite high because you don't know exactly whether there were other processes during the life of the plant that led to differences in the development. But that could be one way. To do it would be to try to parameterize your model at young stages and then run it so you can extend it later on.
Interesting. Very good food for thoughts.
For thoughts, yeah.
So let's continue, and we still have a few
Sure.
more questions saved for the end. Thank you,
Yeah.
Guillaume, and let's keep going. You share the screen.
So the screen is shared again. Check. Read it. So I'm not sure whether the screen is shared or not. Okay, we'll assume it's shared. Okay, so what I want to do now is actually the same type of exercise that we did before, but with another model that is very similar to the previous one. It's actually built on the previous one, and that model is called Marshall. So Marshall is basically a model that includes this relation that you see here. Okay, so Marshall has two components, one which is actually cRootbox. So Marshall uses cRootbox to generate a root system. And then on top of that root system will compute the root system conductivity. To do that, it will assume some values for the local conductivity. Okay, so you can actually change these local conductivities that I showed here. So the radial conductivity, the axial conductivity, you can change them in the modern Marshall. I will not do it here. Okay, I will here focus on the architecture. I will come back a bit later to these conductivities. But bottom line is you can simulate an architecture, and on that architecture, you can get the global root system conductivity that we call KRS. And that KRS is a proxy for the capacity of your roots to take up water. Again, Marshall is fully open source, and there is a website. It's actually an R package, and there is a web interface that looks like this. So, as I said, it's based on cRootbox. So you can actually simulate any type of model, any type of plant. If you can simulate it in cRootbox, you can use it in Marshall. Again, because Marshall is basically putting the function on the given structure. Here in this interface, there are actually two types of root system preloaded. There is a model of maize that looks like this, but that's not the one I will use right now. I actually just click on this button here and use what is here a simplified root system. Just 3 root types, a taproot, long laterals and short laterals. So first order laterals and second order laterals. Okay, I'll just use that, and I will change some of the parameters. You can see here that it's actually the same interface than for cRootbox. As I said, you can also change here. These are the conductivity curves. So the conductivity of a root change along the root axis. We will not change that. You could do it, but I will not do it here. The type of information that you can display here is either the root system, but you can also look at the standard uptake fraction, which is where the water is taken up by the roots. You can look at the axial fluxes, which is here. You can see it's much more important into the main roots than into the laterals, and so on. What we look at here is this tab here, which is the simulation evolution. The simulation evolution, if you actually run the model, here we'll go here and move that. Update the root system. Again, it will just draw an extra point here with the changes in this case, root conductivity from one simulation to the other. Okay, here there are no changes because the root system is the same. I didn't change any parameters. What I also did here is actually put all the deviation to 0. So when you simulate two root systems in a row, it will actually give you the exact same total length. Okay, so unlike the previous one, where I did not put all the parameters or the deviation to 0, I did it here, but only for this simplified root system. But that means that if you look at the total root length here, 1,208, if I rerun it, it's actually the same. It stays the same. Okay, you can see that it's actually different simulations. It's actually different root systems. Okay, there's still stochasticity, but here just in the placement of the roots within the soil domain. We'll just look at the root type again. But so in that case, we will look at that. We will look at how does this root system conductivity changes if we change other architectural parameters. We can look at the root system conductivity, again, the intrinsic capacity of a root system to take up water. We can also look, for instance, at the root system depth. Here, we can see that here in this case, it does changes. Because in this case, the uptake depth, or for that matter, the transpiration of the root system, which is also computed here, these two parameters actually depend on the specific placement of the root within the soil domain. The root system conductivity is only the root system, no matter what the soil look like, no matter whether the soil is dry or wet, it doesn't matter. The root system conductivity is always the same. The root system, the uptake depth or the transpiration, these depend on the placement of the roots in your soil domain. But we will not look at this one. Again, just look at this intrinsic capacity for simplification here. And what I will do, copy paste, is look at compensatory changes in the root system architecture and how different parameters, again, have different effects and can compensate one another. So really looking at combination of these traits and how combination of traits influence then the functional properties. So what I will do here is I will look at, I will first increase the primary root growth rate by 50%. And then I will look at the effects on the global root conductivity. And I forgot to put it here, but also look at the effects on the total length of the root system. And then after that, on top of this one, what I will do is increase the primary root's interlateral distance. So the distance between two laterals, I will increase that by 2 as well, or by 50%, not by 2. And then again, observe the effects on the total length and the effect on the conductivity. So here, So first what I do, and again, I'm choosing these parameters because these are parameters that you can measure experimentally. So these are parameters that you can measure from a rhizostron experiment, for instance. So here I'm changing the growth rate, increasing that. Okay, 1 point, so it was around 1, so 1.6 now. If I look at my root system, it will be longer. Okay, it goes deeper into the soil profile. And actually, I forgot to look at the. So, no. We'll start again. Sorry about that. Here, total length 1,208 for the total length. So I increase. Root system now is bigger, 1,988. And if I look at the evolution, I can see that that led to an increase in the root system connectivity. So that increase in the root system connectivity is due to the fact that I have more roots. If I have more roots, the capacity to take up water is bigger. Okay, the intrinsic capacity is bigger if you have more roots, all other things being equal. What happens now if I actually increase the interlateral distance? We look what it looks like. So I go from 1 to 1.5 in the case. That means that I've increased the distance here between two laterals. It means that altogether I actually have less laterals. So my primary growth rate here, my primary root here is longer, but I have less laterals there. Again, if I look now at the total length, I'm back to a total length that is similar to the one that I had before. So it's not 1,200, but 1,300. So more or less back to the same one. And without surprise, actually, my root system connectivity goes down again. Because in this case, the root system connectivity is closely related to the total root length of my root system. And so what we can see here, these are not exactly the same values because these are different simulations that I did before the one that I just show you now. But changing here, so going to that point, to the first one, to the other one, led to a 30% increase in the root system connectivity by increasing by 50% the growth rate of my primary root, which led to an increase in the total size of the root system. But if you measure only that, and for instance, you did not notice that there was in here actually less laterals basically in your root system, you might have missed that this increase that is induced by the increase in the growth rate of your primary root, this increase might be compensated by a decrease in the number of laterals that you have, or an increase in the interlateral distance in that case. Okay, so that's one example of compensatory factors. It's an easy one. Okay, I chose that one because I knew it would work. There are always potentially compensatory factors between compensatory effects, between different factors of the root system. Okay, so you might say here, okay, that's nice, but in this case, actually, what I have is I'm actually, if I would have measured the total length of my root system, I would have been fine. I mean, these are actually the same length. So why actually bother looking at precisely specific growth parameters in this case? Well, that's because it's not always the case. Okay, I will show you another example that changes a bit the type of interpretation that that you might do, and also see that some type of increases. So from this case, we have an increase. Let's say 50% in the total size of the root system led to an increase in 40% in the total root system connectivity. We can have actually it's not a linear relations in that case. And that's what I want to show in this other example. but we will actually look at the maximal length of the lateral roots. Okay, so I will go back to my initial parameters, and what I will do is go to the lateral, so not the long lateral, but the lateral, and increase the maximal length, go from 2 to 4. Okay. In that case, again, you can see that we seem to have a root system that is much longer because these red roots are much longer than before. And indeed, we went to 1,200 to 1,800 in that case. But if we look at the evolution here of the root system connectivity, you can see here that the root system connectivity has increase of only 50%. So we have an increase, and that's what we see here. We have an increase of the total size of our root system of plus there 50%, which is again very similar to the increase that we had here. We also had more or less an increase of 50% going to that point, then from that point to that point. But in this case, that led to an increase in 40% in the root system connectivity, while in this case, we have an increase of 50% of the root system connectivity. The reason for that is that the different roots don't have the same uptake capacity. And so varying, so if you have longer, in this case, longer laterals, so these second order laterals that you see here, that actually leads to a lesser increase in the uptake capacity of your root system in general, because they have, increasing them, they have a lesser capacity to start with to take up water. So these ones have a lesser capacity to take up water than the other one, okay? Again, what I mean here is that you can actually have, if I'm comparing this point here, 1,800, with this point, very similar length in both cases. If you would have measured only the length, total length of your root system, you could not extrapolate from that length the functional properties of your root system, because again, the functional properties takes more aspects into account and not just only the root system connectivity. Not only the total length of your root system. Depending on which roots are longer and which roots are affected by one parameter or the other, you can end up with different types of different effects on your functions. And your functions here, that is only the root system. We can look at other aspects, transpiration, trying to reconnect. So here, we will again look at the first parameters that I looked at, which is the growth rate, so increasing growth rate of my primary roots, but looking now at the transpiration. Also, this intrinsic capacity to take up water is not always linked to the transpiration capacity or the actual transpiration. In this case, because in between, you actually add the soil on top of that to complexify your system somehow. Okay, moving on. So what I wanted to show here again is that the model in this case, if you have phenotypic data, structural data, you can use that data into a model that can then actually predict the function. And from that, and then the model will actually tell you whether that's the differences that you see in your phenotypic data, whether it has an actual functional differences. Again, you might have, in this case, if you look only at your structural data, you might say, okay, I have an increase in the growth rate, but then also a decrease in the number of laterals that I have. Why do I have that? One reason might be that actually these two plants are functionally equivalent when it comes to water. They have the same function. Okay, these root systems, although they are different, they do have here similar total length, but they have different architectures. In that case, they actually have a function that is similar. On the other hand, if you look at this one, and so 1,700, and this one, 1,800, similar total length, but very different functions, okay? And the way to access that is, again, through the model that can serve as a predictor for that function. And again, coming back to the question, that's possible if the model actually has been developed for that specific function. Okay, moving on to another example, which is anatomies. So far we've talked about architectures. I will just give you short examples about anatomies, which is also some of the work we've been doing in the lab. So it's basically going one step down. So the anatomy is these cell arrangements, is the arrangements of the cells within the root system, and roots have very specific anatomies. With on the outside, you have this epidermis, then you have the cortex, then you have Casparian strip, which is this hydrophobic layer, and then within that layer you have the vascular tissues, so the xylem that will transport the water. And taking measurements of anatomical features is not that difficult. It depends on the plant. If you have very small plants, very small roots, rice, for instance, can be tricky. But if you have large roots, such as maize, which is the case here, it's actually not that difficult. So what you see here, these illustrations that you see, these are handmade sections of maize roots. And what Adrien did, so Adrien Heymans, we did this study, what he did was to actually make cuts along the roots. Okay, he made cuts at different stages along the roots and wanted to see whether we could make a link, how the differences that we would observe in the anatomy, how that will translate into differences in the function of the different roots along, again, the root. So what he did was cutting, making cross-sections, looking at these cross-sections under the microscopes, quantifying these cross-sections, which means quantifying, looking at the number of cells, number of cell layers. Just zoom in. So looking at the number of cell layers that we have here, the size of these cells, the size of the xylem here, the number of xylem vessels. You can see that we have 2 here, but we can have 3 later on, and so on. And then with these type of phenotypic information, you actually use a model that is called Granar, which is a generator of anatomy. So Granar, what it does from anatomical features, it regenerates a synthetic version of that anatomical network. Okay, so we have here, what you see here is always a computer-generated anatomy, but with the specific data that were acquired here with the cross-section. Okay, so here, basically what we have, the cross-section, we extract the data, we use that data as input for the model, and the model gives us this. Okay, so this is very nice. Okay, but this is, at this point, this is just an illustration. Okay, it's just for, it's just showing nicely the differences in anatomies that we have. The good thing is we actually have in the lab someone developing another model that is called MECA, And MECA, it's a micro-hydraulic model, which is basically the same principle as the water. It works with the same principle as the other one I showed before. So it computes water flow based on these physical relations, these physical equations of flow between compartments, but on the whole route, on the whole cross-section. Okay, so instead of having different routes, you actually have different cells, and between these different cells and cell compartments, you simulate the water flow between all of them. Okay, so you can have the flow between one cell to the other, the flow also between the symplast and the apoplast, for instance. Okay, again, I will not go into the details. The good thing is, as soon as you have one of these cross-sections, you can actually estimate, again, the hydraulic properties of that cross-section with this other MECA model. So if you input this model, this anatomy generated with a granar into MECA, then you can get radial connectivity and eggshell connectivity, for instance. So we're not talking here about the connectivity of the whole root system, but local connectivities. Oh, and that's, I forgot I had that slide. So that's what is showed here, that MECA, again, can compute, can compute water flow between all the different cells, fluxes between the different compartments and so on, and estimate the radial and eggshell conductivity. And so if you combine that model, and for every root section that you actually had experimentally, so again, you have your experimental root cross-section, you extract the data, you generate a synthetic version of the data, and then from that synthetic version, what you do is you compute the radial and eggshell conductivity. And that's what you see here, is actually you see the evolution of these functional properties, local functional properties, eggshell, radial conductivity to water. You can see their evolution along the route, again, on the data that you have here. So these functional curves here, okay, where you can see that we have different curves, each curve is actually for a specific root type. They are coming from this model and from this model here and coming from this experimental data. Okay, so this is another example where the structural data can be extended to functional properties. All of these, okay, experimentally, it's very hard to measure this type of data. So these radial and eggshell connectivity are very hard to measure experimentally. What we can say here is that they always fall within range of published values. Okay, so that makes sense. These are not random data that do not make sense. They make sense, and they are within range of experimental previous experimental values. Okay, so that was a second example, but with anatomies, as I said, we not go into too much details here about, and we actually don't have web interface that link these two models. We do have a web interface for the models. So if you go on the GitHub page, the reference, you will see that there is a web interface for Granar, there is a web interface for MECA, but not for the link between the two. The thing is, in this case, MECA that you see here takes actually a long time to run, and so it's not handy to include it into a web interface. What we did here, what you see here is actually pre-compiled results. So you can actually test different type of routes, okay, the different complexity of routes, but all the results were pre-compiled. So the model is not running into the background, which is why you not actually attach these two. MECA, on the other hand, you can actually change directly the model in the same way. You can change the different parameters of the model, changing the number of layers of the cortex, the steel number, the number of xylem vessels. If you want, you can change basically everything you want, but in this case, it only shows the functional aspects and not the structural, sorry, not the functional aspects. Okay. Last part in the workshop is, well, this is nice. I would be actually interested to test that, but then how do I do? How do I actually move from data into model parameters? How do I move from my phenotypic data to these functional insights I can get with the models? I actually advise that you should first of all model that you're planning to use. Okay, so for that, first know the question that you want to answer. Depending on the question that you want to answer, you will then find models that are fit for that specific question. Okay, and once you have that, so it starts from your question. So the data that you are, the global aspects that you are interested in, then you choose the model that you want to work with. And from that, then you look at, okay, what type of data do I need to get to the model? The model will define the data that you need to take. Okay. If you want to have an idea of the type of models that exist, you can go, and I think I put the link here. Yes. So on the website. So that's a website which is just referencing different types of models that exist. Okay, so these are not all models that we develop in the lab.
Excuse me, Guillaume.
I think they are
your
now...
connection, sorry, I apologize for the interruption. Your connection seems a bit dropping in and out. Could you just go back to the beginning of this section a bit and start it again?
Okay.
Yeah.
Is it better?
Yeah, we lose a bit of few words here and there, but sounds okay now, I think. Oh, we apologize for the inconvenience. It's the things of the online workshops.
Yeah,
Guillaume, are you still with us? Guillaume?
I think it's the video that has on the presentation that is slowing down a little bit.
Yeah, I suppose these platforms are quite heavy, so they kind of take a lot of the internet. So we are working with Guillaume in solving that. Meanwhile, I'm gonna post. We are gonna post in the chat. Guillaume?
Yes.
Oh, you're back. Sorry, I think
I'm back.
When you connected to the platform, we lost you for a second. I think it was taking a lot of your Wi-Fi. So if you would like to kind of...
Yeah, I will go back. Okay.
Excellent.
So,
thank you.
Apparently I had too many windows open for the
That's
the
one of the
bandwidth that I'm
downsides.
working with. Yeah. Okay.
Thank you.
Right. Okay. So I will actually not show. Then I will go back to this point. Sorry if you heard half of it. So how do I go from the data to the models? Basically, that's the question. If you are interested with using the models, what should you do now? And how can you actually adapt your phenotypic data to connect these two? My advice is that you actually choose the question first. So you should know exactly what you are looking for. We discussed that just half an hour ago. If you are interested in the structure of the soil and the effects of the structure on the water retention capacity, then you should look for a model that does that. Okay? So define your question, and then once you've defined your question, then you define your model. What I wanted to show you, which I will not do again here, is that if you go to the GitHub webpage, there is a link to the plant quantitative website. That website is actually reference 100 of models, plant models, and you can filter out by processes and so on. And so if you're interested in nutrient uptake, just select that in the drop-down menus, and then you can look at what is out there. And then once you have your model, again, if you're not sure, don't hesitate to contact the contact person for these models. Usually, people are very happy to answer your questions about their tools. But then once you've chosen your model, then what you need to do is to look at, okay, what are the inputs of that model? I will actually put here more generally inputs, okay? So what are the inputs of the model? And once you have these inputs, you look at, okay, we'll put input again here. What type of experimental setup can I use to get these inputs? Okay, if your inputs are, I don't know, just the total length of your root system, what type of experimental setup will allow you to get that measurement? Again, the problem with roots, or the challenge with roots, is that more often than not, you actually don't have one specific platform that can do everything. Okay, or every platform comes with a set of parameters that you can extract it from. And so for me, that would be really the way to do it, to start actually with a question, and then the model that can answer that question, and then from that, design your experiments. So from the model, look at the inputs of the model, and from the inputs, which experiments can be used to get these inputs. We've done the job for cRootbox. Okay, so I will show you here. Where is it here? So if you go to this digital root worlds database, hopefully that should not crash my computer, my connection this time. So this is actually a database of model parameters. Okay, so it's a bit wider. So when we went to the cRootbox interface, there was actually a set of parameters there already pre-entered into the interface. The aim of this other interface is really just to reference all the reference model parameters. And we have so far 3 models. So it's very much still a beta version. Okay, it's not fully as we want yet, but we have more than 1,400 parameters, 58 full parameter sets for 3 different models. Okay, so you can just browse that if you want. It might be useful if you're looking for, let's say, parameters. You want to work with cRootbox. You want to select, want to work with, I don't know, sunflowers, no specific genotypes, and you want to have an ID for the data for the primary roots. There we have already values for the different parameters for the primary root, okay, for sunflower or for cabbage, if you're interested in cabbage, okay? And so we have that for cPlantBox, ArchiSimple, Granar, and Meka. I forgot we had that one. No, there is nothing there. Just Granar, ArchiSimple, and cPlantBox. But that's actually not what I wanted to show you exactly here. What I wanted to show you is this data network tab, where, and what we did here is actually link cPlantBox parameters. So in this case, it's only cPlantBox parameters with specific platforms. Okay, so if you click here, and again, this is a beta version, so here it's the abbreviation of these parameters. But I will take R, which is the growth rate. And so here, I will remove that one. I can see all the different, theoretically, the different type of experiments that I can use to get these growth rate values. Okay, so for instance, what you can see, Chauvelomics, you cannot get an idea of the initial growth rate. Why? Because it's actually destructive measurements. Soil coring, you cannot either, because to have growth rate, you need to have different time points. Okay. Excavation, trenching, you cannot have an idea of these. You cannot get these type of parameters. I would take another one. I would take, for instance, here the LN, which is in that case, the interlateral distance, I can get it from excavation. So if I excavate some roots, I can get that from, I can get these interbranch distance. Okay, so that's just an example. Again, I will not spend too much time here, but to show you that you can actually link specific phenotyping techniques or phenotyping setup with specific types of with specific parameters. We've done it partly here for the plant box. You have to check whether that has been done for other type of models. The idea here would be that at some point there will be actually that, but for every type of every type of model that you might want to use. We can also do it the other way. So here I just selected to do by the methods. So I can say, okay, I actually have a rise-drawn experiments. What can I measure with a rise-drawn experiments? Okay, can I measure everything with a rise-drawn experiments? No, there are some things that you cannot measure actually with a rise-drawn experiments in that case. Okay, so that was just one example. And then to conclude, what I wanted to show you is quickly also a small demo of a tool that we actually developed again in the lab that was more than 10 years ago, which is called SmartRoot. So it's an image analysis tool for root images. And I want to show you that if you actually have SmartRoot data, from SmartRoot data, usually you can actually extract quite a lot of information that can be directly used with the type of models that we have. So SmartRoot, if you open it, it actually looks like that. Again, I'm not going into the details on how to install it and so on, because that's the information is, there is a link in the GitHub repo with the link to the SmartRoot website. But what SmartRoot does is, check that. So here, what SmartRoot does is actually not working. I'll just restart. Yeah, so SmartRoot is a plugin of ImageJ. So it's very easy to install. You just have to install either ImageJ or, in this case, Fiji, which is a distribution of ImageJ. It comes as a plugin, and SmartRoot allows you to trace. Oh no, that's the one. Trace different types of roots. Okay, so that's what you can see here. So this is a lupin root system. Actually, I pre-traced most of the roots here. Okay. But I will show you on a root that is not traced. If I left one on another, like this one. Not that one. I will delete that one. So it's a semi-automated root tracing tool that allows you to trace roots. So see here, that was done semi-automatically. Again, we'll not go into the details, but you can see that that root was fully traced directly. This is not a great image because the resolution is not that great. What we try to put in SmartRoot is actually try to capture most of the information that you would need to characterize a root system. In this case, you can see here we have actually different types, different colors for the roots. We have the green ones and the yellow ones. That's because the yellow root is known to be the primary, and all these green roots actually SmartRoot knows that they are attached to this primary root. So coming back to what I said at the beginning, SmartRoot knows the topology of your root system. SmartRoot also knows the length of the different roots because it has traced the different ones here. Again, this is a bad tracing. I just did it very quickly before this morning. it's not done properly. But if it's done properly, then you can get the length of every single root. You can get also the diameter. You can see here we have the diameters of the different roots. So it contains a lot of information, topological, morphological, even dynamic information if you want to. And that information is stored into a file. So if I close it here, that information is actually stored into an XML file like this one. Okay, that contains all the points in my root system. So it contains again all the different roots and all the different points of the different roots and so on. This file is called an RSML file for root system markup language file based on XML. Okay, so we have all that stored into this specific format. And then what we can do, we can actually, once we have traced the different roots, we can actually export the data from that format, that RSML format, into a table format. And so we can here, I will do what is called here in SmartRoot a batch export, in which I will select a folder that contains all my root tracing. In this case, it's directly on my desktop. There is only one here, okay? There is only one RSML here. And I will ask SmartRoot to export that into a CSV file. I just have to choose where I want to put that CSV. And I will put it on my desktop as well. Here, okay? I will just delete the previous one. And then once I've done that, what that will do is that SmartRoot will look into that folder and open every single RSML file into that folder, and then export the data for that RSML file into a single CSV. Sorry, just for the demo, what I will do is I will just copy paste that one several times. So now I have 4 RSML files. They're exactly the same, but it doesn't matter. And then you just run here, the export. Now it's done. And what we can see, and that's the reason why I wanted to show you that, is that first we have the data from all the image. So we have the first image here, and then we have, where's the other one? Oh yeah, so we have 3, 3, 3 here, 3, 2, and 3, 1, I guess, at some point, 3, 4, and so on. So we have exported the data from every images that we had in one single file. And then for every single root, what we have is we have the length of the root, we have the diameter of the root. If the roots, I will just try to zoom in so you can see better. You can also have the density, the child density, so the number of roots that you have per centimeters, which is directly the inverse of the distance between two laterals. You can also have the position of the different laterals. So basically, again, I will not go into the details, but once you have this type of tracing, either with SmartRoot or there are other tools that can do it as well, you can actually extract from this type of data, you can extract then the parameters that you need from for the models. Another thing also is that, and I will conclude on that, is for some of the parameters, you actually don't need to trace every single root in your image. Again, coming back to the idea that tracing the whole root system to get the total length might not be always useful or possible, or it would take too much time. However, in this case, if I want here to estimate the distance between two laterals, I actually don't need to trace, I might not need to trace all the roots in my root system. I might just say, okay, here, for instance, here it's a mess. I don't see all the roots. They seem to be going back and forth. So this is too messy. Okay, I'm not sure about the quality of the data in this situation. What I will do is I will actually go back up here and say, okay, here in this region, I'm actually more confident about the data that I can trace. And I can say, again, not going into the details here, but what I will do is I can sample my image and say the density of roots, the lateral density, I will actually get it from that area here. And I will maybe get another sampling in my root system, maybe go here. Okay, here I have another sampling because here I can see the roots. Here it's too messy. I don't see them. I don't trust that part. Or here is another sampling that I can trust, and so on. What I want to say here, again, without going into detail, is that for most of the parameters that you can get, that you might need for the models, you actually don't need to trace the whole root system. You don't need the data of every single root. You can actually do sampling of your roots within your root image, and from that sampling, get the data that you are interested in. Okay, so to conclude, coming back to the what I said at the very beginning, I hope I showed you some examples on how models can help you extend phenotypic data into more phenotypic data. So the model can serve as kind of analysis tools that can extend some data into more of this data. Get data from a young stage, run the model, and see how that data at the young stage translates into data at later stages, for instance, or how changes in the growth rate of one specific root will translate into differences in the total root length, for instance. That's one thing. The next one was how can changes in the structure will influence my function? And we've seen the example with the water uptake. And then how these from different structures we have different functions, but sometimes from similar structures we also have different functions, or for different structures we have similar functions, and so on. So basically the model can help us extrapolate structural information into functional information that we might be interested in. And finally, my general advice, I was not very specific here, but my general advice on how to link experiment and model is actually start with the model. Start with the model because the model will answer a specific question that you know your question, you're doing your project, you know your question. Find the model that can answer that, and then from that, look at the data that you need to run that model. And from the data, look at the experiment that you might need to do to run that model. And that's it. Happy to take questions for the final 7 minutes.
Thank you so much, Guillaume. This is a fascinating world. Thank you so much for all these fantastic resources, for letting us know about them and going through them one by one. That's really great, and I'm sure you will get. If that's okay with you, we will share your contact details because there was some interest in.
I mean, yeah, my contact is normally not very hard to find, though. So
not very hard to find. No, it's very hard to pronounce, though.
Yeah. Okay.
Well, I actually, we still have a few minutes. Normally we don't like to spend it beyond the 2 hours, but I will. We wanted to ask you kind of a little bit of a favor to summarize a bit some of the main points that actually for the general audience come, like with the importance of generating this, collecting and collaborations to collect these data sets to actually develop these models, which bring me to the second point. The importance of this model is also like to make the most of your data, because sometimes these phenotyping experiments are expensive, are time-consuming, and the possibility of developing further parameters beyond just your measurement of a few morphological traits to actually predict other functions is definitely really important. I would like you to highlight this from this workshop as well.
Yeah. So once you have the model, as you said, the hard part is getting the data. Definitely, and that's partly what so there is a real value into finding that data, generating that data, and making it available. That's why with the database that I quickly showed, that's the idea there. It's not to share phenotypic data, but share model parameters. Again, because these are usually hard to get. And we hope to add more and more of these, update that database so we have more and more parameters for more and more species, so more and more people can actually use it. For me, the value of this type of database is that even though they might not correspond directly on your specific experiment, let's say you do mice, and in the database you find a data set for mice, but not the right genotype that you're using, but still you can reuse that. The idea is that you could reuse that data set and then just modify it. You don't have to start from scratch. You modify something that is close to what you want to do and then reuse it there. Does that
more and more projects are encouraged to share all this data. So sometimes you can find fantastic data sets from previous projects.
And if something we also started to do a few some time ago, if you go on the quantitative plant website, there is actually a tab for datasets. So you also have their link to existing datasets, but in this case, it's more image dataset than really dataset themselves.
Maria, had a question for you too.
Sure.
Yeah, my question actually is, how do you see these models, the application of these models in plant breeding?
So in plant breeding, for one, different things. There can be a guide for plant breeding. So one thing that I showed at the beginning, no, actually I did not show that slide because that was a bit out of the topic. So when I show the slides with these thousands of simulations, so we had specific route, so when you combine the root architecture with the functional, so when you use the model Marshall, you can actually do a lot of different simulations, but varying all the different parameters. So that's what we call doing a sensitivity analysis. So we have our models, and you just try all the possible, you try many combinations of traits that you have, okay? Possibly much more combinations that you could try experimentally. Sometimes just because these combinations actually don't exist in your gene bank, for instance, or your line collections. And then once you've done that, and that's the part that I did not show, is that you can actually do what is called a sensitivity analysis. The sensitivity analysis is looking from that huge data set of simulated data. What you do is you actually make the link between the inputs and the outputs, and you rank the inputs, and you say, okay, that specific inputs, the growth rate of the primary root, is the one that will influence the most my water uptake. Okay, and because you tried all these different combinations with your model, and because that's a mechanistic model that really takes into account the physical processes, then again you can evaluate you can rank the different parameters by importance, and you can evaluate the contribution of each parameters. Yep. You could say that parameter, growth rate of my primary root, is consistently improving my water uptake capacity, for instance. So then that's, for me, that's one way of guiding breeding exercises, is to look for these important traits.
Yes.
It can also more generally be a guide for phenotyping, because if you're interested in a specific process and you want to see the differences, the model then in that case can help you just define what you should look for. Maybe looking at tropism is not important in that case. but it's important to looking at other traits.
No, thank you for clarifying. It's very good.
Thank you. I have just a little technical questions. Where, I mean, in like to develop this model, obviously you need very quality data. Is it possible? Do you think it's possible to develop it also from field work, from field phenotyping, or they are still limited a bit to lab phenotyping, which is obviously much more controlled and.
Yeah. So here we talk about specifically root models.
No, roots. I mean, because roots are particularly difficult to.
To get into the fields. Yeah, getting the shoot part into the field is not more difficult than getting the shoot part into the greenhouse. So that's an aspect that is still very much active. It's a question that is very much active in the community, and that we try to answer in multiple ways. One way is to try to find proxies, find parameters that are easy to measure in the fields and that you can then extrapolate into the models. But that we don't have any real convincing things. What is more often done is what is called inverse modeling, where what you simply what you would do is, for instance, do a coring into your field. Okay, you do a soil coring and you get a profile of the length of your root system with depth. Okay, so you know that you have that amount of roots at the top and then it decreases in that way as you go down. Okay, the thing is that's actually the output of your model. Okay, of the models that I showed, this depth profile, that's the output of the model. And so the way then you can use the model in this case, because you measured the output of the model, what you will do is you will change the input parameters up to the point where you reach the same output as your experiment. Does that make sense? So you try a lot of different combinations for the inputs, and when these different combinations, when the output of these combinations is the same as the output of your experiment, then you say, okay, these input parameters corresponds to what I could have measured into the field. Okay.
Some sort of validation.
Right, if you want. The problem is often with these very integrative measurements, such as the depth profile, you have different set of parameters that can lead to the same profile, basically. And so these inverse modeling exercises are actually not easy to do correctly. And we're still discussing how to do it in the best way and how to do it in a predictive way and so on. So they're not, roots in the field, that's still very much the challenge today.
It is a challenge today, but it's it's what we're after at some point. It's the next
fully agree. We have to go into the field. And I truly believe that the models can help us do that, but we are not there
exactly.
yet.
Exactly. That's what that's what the thought of many. We have reached 5 p.m. It's Friday. Well, 5 p.m. in Europe, and it's still a long day. At the other side it's a long night. I think I would like you also to join us in giving a bit of a message that all these, particularly these models, are giving us the idea that root phenotyping is not just for plant physiologists, but it has a tremendous importance for crop scientists, environmental scientists, and soil scientists like myself, because now we can connect all this data into much more in depth. And I think that's also reflected in the audience. I mean, from all the attendees we have registered, the variety of interest is quite broad. So it's a good reflection that root phenotyping and plant phenotyping in general is just way beyond just a plant scientist. And with that said, we want to thank you, Guillaume, for your time today and all the interesting and fantastic information you have shared with us.
You're welcome. Thank you.
And we are going to have a little break again because it's a lot to think and process with these workshops. I imagine you will need to come and revisit us. We might have one or two more workshops in December. One of them, I'm posting the link in the chat. It's about us, yet another free resource. It's called For the Root. And we are pending to confirm the workshop with the speaker. So please keep an eye on Twitter and LinkedIn and all our social media, because when it's ready to be announced, we will be making as much noise as possible as usual. And there will be a last one, quite possibly by myself, in which I would like to touch something that we cannot escape in root phenotyping, and is the impact of root exudates in microbes. That's something we have been we are still also, like the field work, we are still very much starting to realize it's a functional phenotyping, as we tend to be told. Again, this will be announced. It will be in December, so keep an eye on our media. And thank you so much for joining us today. Come and rewatch the video anytime. Thank you, Guillaume, and see you soon. Bye-bye.